Good evening, everybody. And thanks to the tech wizards for setting this up. Are you on record? You are being recorded. This meeting is being recorded. Welcome to the 1241st regular meeting of the Entomological Society of Washington. I'm your new president, Don Weber, and the first order of business is the reading and approval of minutes with Secretary Gary Cullen.
Thank you, Don. Good evening, everyone. The 1240th regular meeting of the Entomological Society of Washington was called to order on the 7th of December by President Matt Buffington at 7 p.m. in room WD 33 of the National Museum of Natural History. 21 members and 3 guests were in attendance, with 19 virtual visitors. Recording Secretary Gary Hebel read the minutes of the November meeting, and they were approved without corrections or comments by attending members. The annual December meeting is also a business meeting. President Matt Buffington shared information from reports of the society officers, and these reports will be published in the proceedings of the ESW. David Adamski, leader of the Young Entomologist Group, was acknowledged by President Buffington for his past and present activity. David first noted that YEG had an annual business meeting recently at Huntley Meadows, Virginia. A new slate of officers for 2024 was presented by President Buffington, which included Abigail Kula as treasurer, Gary Hebel as recording secretary, Alan Norbaum as program chair, Andrew Jansen as president-elect, Talitha Simoes as curator, Jerry Cook as co-editor with Mark Metz, and Elizabeth Young as membership and communications secretary. President-elect Don Weber automatically becomes president. This slate was formally presented to the society and was approved without reservations. Show and Tell included a Xerces Society t-shirt worn by David Firth. Dave also noted that the Polish company has emerged as a producer of insect drawers and unit trays. President Buffington displayed a new book, Wasps of the World, by Simon Van Noort and Gavin Broad. The book includes one page devoted to each of the families of wasps. Program Chair Alan Norbaum introduced the speaker of the evening, Dr. Brandon Boudinot, a postdoc at the museum, whose presentation was titled Phylogenomics and the Evolution of the Hymenoptera. Dr. Boudinot is an evolutionary ecologist and has engaged in such work in Germany at Friedrich Schiller. He will return to Germany's Stuttgart Museum in January. His experience has been partly with the evolution of beetles, but now with focus on the formicida. Synchrotron scanning provides very high power X-rays. Such scanning allows all kinds of interpretations. The technique allowed comparisons of body areas in ants that were not previously seen. Locomotion of ants was also studied, and individuals were found to have efficient movement processes. They have spatial comparisons to cheetahs. Synchrotonic scanning provides a new world for comparison of insect structures. The last action during the meeting was a ceremonial passing of the official society gavel, President Buffington to new President Don Weber. The meeting was adjourned at 8:40 p.m.
Are there any suggested changes or corrections to the minutes?
Frankfurt.
Oh, sorry.
Okay.
Also, Andrew Johnson, not Andrew Jackson.
Jansen.
Jansen, sorry.
See how easy it is?
Got one of the vowels right.
So that, Boudinot has always worked on Hymenoptera, but he's moved to beetles once in a while. but it's always been a Hymenoptera, I think, right?
Yep.
Yes.
Who needs it tomorrow?
So noted, right?
Sure.
I think we approved.
Can we now approve the minutes? All in favor, say aye.
So moved.
Motion.
Second.
All in favor, say aye.
Hi.
Motion. Any opposed? Motion passes. Thank you, Gary. It says now reports of officers and committees, and in my first meeting, I don't know exactly how to handle this, so I'm gonna maybe rely on past President Matt Buffington to guide me. Do we call on each of the officers?
I usually
or-
take a look at what officers are here
Yeah,
and then ask them or see who's online.
and I must say Andrew Jansen, President-elect, is not here. So, do any of the officers or committee heads have reports or business to bring for us?
We received the last proceedings. It
Right.
arrived, yeah.
Okay, and Lourdes, were you gonna, you said you were gonna post that on the website?
Oh, we might send out an email to the membership with the table of contents. And that's something that we're thinking that might be nice for the membership. Once the new issues are published.
Yeah, that looks really good.
So we're working on that.
That's what volume 123, number 1. And it's really, there's some really cool articles in there. In fact, I'll point out at this point that one of the articles on the old arboreal ant colonies was featured in the Entomology Today, which was a sister society, Entomological Society of America. And there's a link to ESW, proceedings of ESW, and also a full YouTube. Where she's up in the trees.
Also, a substantial article on Anastrepha.
Yeah,
Post plants. And stuff.
And Zach's first article as well.
That's right.
Yeah, so it's nice to see them.
I want to call now for the introduction of any visitors or new members. Yeah.
I'd just like to point out that my new employee, Dr. Jeremy Frank, who was not here when we met in December, he actually came right before Christmas. So this is his first meeting and hopefully not his last meeting.
All right. You want to say any words?
Yes, well, membership. I still need to actually pay you, but I will.
All right.
So I'm a research visitor at Smithsonian and I came to the talk.
And what's your name, please?
Oh, sorry, my name is Emilia Albuquerque. I was a fellowship here in entomology. I
Excellent.
also work with ants. So.
All right. Need a lot of
Or too many camp.
Any others? And yeah, online.
Does anyone online want to say hello?
Do we have any visitors online who want to identify themselves? And nobody's raising their hand. Okay, well, they're quiet. All right. And do we have any new members? Let's see. All right, soon, right?
I do know that there were a few new members, but Elizabeth Young is not here. But I do know that we have a couple of new members through the website. So we'll read them in the next meeting. But and then just to remind people that you'll have automatic renewals coming for those who signed up through the website. So just to remind you about that.
Don't panic.
Don't panic if you see that charge. Well, I'll ask Mr. Past President to tell me if there's any unfinished business. Of course there is, but any unfinished business to bring
Well, I don't know what to categorize this as cookie.
Yeah.
Um, Back in 2014, I guess, there were T-shirts made up for the banquet. And anyway, Sheila Marita came
This
by
is-
this week and picked up some of her
To
stuff.
clarify, this is 9 years.
You mean this T-shirt?
Anyway, she came by, because she's moving to California anyway, and she had found this box, so it has some T-shirts. And it has uncashed checks from Dave and Dave Firth. You have these back if you want.
Unfinished business.
But I thought maybe you didn't get your shirt, so I. 2015.
I'll buy that shirt
There's
from you.
nobody gonna.
If anybody else wants one. Some here. There's a few more.
Oh.
You can fight over them. But Don,
You try and cash these checks.
No, that's.
Some more in my office. if anybody really wants.
Thank you, Alan. This is truly unfinished business. That was left hanging. Okay. Maybe we can go back to those people and have them rewrite the change.
Yeah, I don't know.
All right.
Do we have any new business? Okay, I'm gonna quickly go to presentation of notes, otherwise, and exhibition of specimens, otherwise known as show and tell. Alan, you had something?
Our former very active member, Chris Thompson, passed away a few years ago. There was a Pestra Memorial volume published in his honor. It's really nice. There's a really nice obituary, various dedications to him. It's effect on serpentology, etc. Quite nice. Pass that around.
It's, what's the publisher?
Oh,
in
and,
Germany?
oh, Tina Litwack, the cover art. Some things, this is actually your copy, Tina.
It says
Oh,
so.
yeah, that's right. I should-
There's your name in it. so.
Don't have an eraser?
To dipterology and other.
Oh, I don't.
The publisher's in Switzerland?
Wow, this is really neat.
I won't say Germany. but-
Germany, okay.
Sweden.
Unless
Somebody else
it's changed, there
any-
was a small box of those in,
Torsten had gotten.
I don't know if he's distributed.
Any other show and tell? I have some tell. And it's along the lines of books. I am the book review editor for American Entomologists now as of the first of the year. So if you have items like this, or if you already have them in your hand, or you know of them being issued, and you want to do a good review for American Entomologists, guess what? You get a free copy. If we can arrange that, you have to do it through, if you do it through me, I request, and you get the book and it stays on your bookshelf as long as you complete that book review, then it's a good one, right? So yes,
What about edited volumes?
edited volumes are fine. It's more a matter of, is it a significant work that's of interest to entomologists in general, or tourists, or et cetera. So certainly, and bring it to my attention. And just on where we're at, that's pretty easy, right? But please, we're always looking for it. There's two categories really. There's the ones that get a review, and I can give you the specs on that. And then there's ones just immediately when we find out about it, says, received, quote unquote, or basically we heard of. And those often in the next quarterly issue would get reviewed if there's enough interest in someone to do. So I encourage you to do that. I also want to acknowledge that Mary Cornelius actually the Invasive Insect Biocontrol and Behavior Lab, ARS, at Beltsville, which I'm a member, passed away on December 7th. It was actually the day of the last meeting. And there's going to be a obituary going out within ARS. I'll also think of one for the American Entomologist. I know that you and Mike Gates and others working on parasitoids work with Mary. If you look at her career in general, she worked the most on termites, specifically Fromos and subterranean termites. When she was with Ken Grace as a postdoc in Hawaii, and then for many years in New Orleans with ARS. So Mary had a long struggle with metastatic breast cancer, and she was a real friend, very kind, quiet, and dedicated. So just wanted to let you know about that. And finally, I don't know if Bridget Bradshaw is online.
Yes.
Okay. I wanted to call it, Bridget is working with the Clifton Institute out in Warrenton. And one of their several studies is studying the food of kestrels, the smallest native falcon. And that group has a lot of insects, which kestrels often subsist on. And many of them are beetles, which are as yet unidentified. And I don't know if Bridget, you wanted to say a few words, but we do have some Coleoptera people in the audience, and also some others who might be interested to suggest how to approach the identification of some of these samples. Are you there? And unmuted. She's got her hand raised. You got to unmute yourself. Do I have to do anything?
Okay, wait, I figured it out. Can you hear my voice?
Yeah.
Okay, great. It's a miracle. Sorry, you caught me. I was like mid eating a bite of food. Yeah, thanks for the intro. So we have, like Don said, we have a zillion hours of footage of, well, he didn't mention this, of footage of kestrels bringing all sorts of stuff back into nest boxes, but we're also trying to match those photos, still photos, with nest box contents. So like bits, weird bits that are in the bottom mixed with all sorts of gross stuff, including a lot of sawdust. And so we're trying to find people, a lot of poor unpaid interns and some high school interns to just separate out bug bits. And then the much more laborious task of trying to figure out ideally 2 species, but really just as low a taxonomic order as we could get, all the different, there's like grasshopper legs, but surprisingly a lot of beetles, which we were not expecting. And so we're sort of collecting them in envelopes right now, and TBD what we're going to do with them. But I've been looking for a good source of where to post photos of things that we have questions about. I mean, we're like an hour out of DC or something. So I don't know if you guys ever have like bug bit identifying parties, but yeah, it's a huge amount of work, and we're not really sure how to get this amount of labor done. So yeah.
Well, I mean, I think there's a lot of good ideas out there amongst this group. I don't know exactly the best idea to connect. Bridget, can people contact you through the Clifton Institute website, or how would it be best?
Yeah, yep. Any of the like if you go onto our website, it'll have everyone's email that works here. There's only like 8 of us, so okay. If you email any of us, it will get to the right person. I'm not the lead on the research. I just like bugs, and so if people find something under the dissecting scope, they're like, "Do you know what this is?" And sometimes it's yes, and sometimes it's no. So, so I've been kind of trying to pass along photos of things. But I'm just sort of starting to collect an album of mystery photos. So if anyone just wants to access to mystery album photo, happy to do that. I've tried putting things on iNat and it just sits there barren and desolate. So yeah.
Thanks, Bridget. We'll prod people to get to your website and,
Maybe we can circulate a link to the iNow photos that are already up.
oh, okay.
Oh, that's a good idea. Okay. That's a good idea. Thank you.
Someone else had. You.
So Tyler Litwack just brought this to my attention. This is a brochure that we've been working on about the invasive chestnut gall wasp and its parasitoid Pterimus sinensis. And these are hot off the press for the invasive species working group meeting next week in Annapolis. So I'll pass this around. And I have some, this is all we have, right? Okay.
We got 200.
Okay, we can, you really want one, we have some here. I just need enough for the meeting next week. And thanks, Tyler, for getting that done. It's great. Nobody left over.
So my show and tell is a little too heavy to bring to the meeting. But for those of you who have badge access in entomology, a number of months ago, Nest Zero of the Northern Asian Hornet, aka the murder hornets, came off of display where it was in the hour places. And yesterday, myself, Desmond Smith, and some folks from conservation built a mini exhibit, which is now on display on the Hymenoptera floor of entomology, with actual specimens, the nest, the log, and it's right, you can't miss it. If you get off the elevator, it's directly in front of you. So
And the suit.
feel free to check that out. Yeah, I got a mannequin. I filled the thing. It's got the suit and everything.
It is a little terrifying when you get off the elevator.
Once I had that thing, I was moving it around the department, but nobody was really around, so it wasn't nearly as satisfying as I thought.
You mean the super suit,
Correct.
the one that looks like an astronaut?
Yep.
Yeah.
Yep.
APHIS just sent
With
out.
the, with the little,
You
with the
know,
little venom shield and all the fixins. So anyway, if you happen to be strolling through entomology, go to the 5th floor Eastport and check it out.
All right, any other show and tell? Show and tell virtual? So now we're ready for the main act, and I'll ask Alan to introduce our past president concerning adventures in the Hymenoptera.
Okay, our speaker tonight, our past president, doesn't really need introduction, but Matt Buffington received his master's degree from Texas A&M University in 2000, mentored by Bob Wharton and Jim Woolley. Returning to his native California, he earned a PhD at UC Riverside with John Parody and John Pinto. Following graduation in 2005, Matt went on to a postdoctoral position with Fred Ronquist at Florida State University. In 2006, he joined the Hymenoptera unit of the Systematic Entomology Laboratory of the USDA, stationed at the National Museum of Natural History, where he remains to this day. Throughout his career, Matt focused on the systematics of Cynopoidea with an emphasis on the Entomophagus Vigidis. Matt is interested in all aspects of Hymenoptera research, from field, bottle to genome. He likes to experiment with novel collecting and sorting techniques, labeling techniques, and scientific photography. Matt also enjoys mentoring students and interns at the USNM and nearby universities, and has also co-organized 7 offerings of the Hymenoptera. Presently, he's involved in collaborative efforts reconstructing the Hymenoptera tree of life, as well as more focused work on species limits questions within the cynopoid wasp and fire ants. Dr. Buffington.
Well, this is a real pleasure and honor to be presenting to you all tonight. And a great deal of fun putting this talk together, because you get to review your career to this point sort of talk. And it's come together in a nice story. And you're all my friends. It's such a, means a lot to me that you're here on such a cold night. So with that, let's just get started. Oh, by the way, 10 was just like an arbitrary number. I think it's like 20 years. I don't know. It seemed like a good number. So let's see if we get this to go. Hide fully control. Ooh. Any idea how this advances?
Arrow, click once on slides.
How about that? All right. That's new. Okay, so there's gonna be 6 kind of components to this talk. We won't be here all night, but I'll keep it moving. You won't have anything to nap. But we're gonna start from the broadest. We're gonna look at the phylogenomics of all Hymenoptera and kind of work that we've been pursuing here since about 2013 to reconstruct a Hymenoptera tree of life that we can really have some reliance on, some intuitive interpretation. That has yielded a bunch of sub-projects, one of which yielded results actually just today. But I'm going to focus on the twig Cynopoidea, which is my main research group. And things we learned from the all family analysis and what it led to our understanding for cynopoids and kind of the novel results we got out of that. Then I'm going to move on to what we can do with a stable phylogeny. Who cares about a stable phylogeny? Well, I do, because with a stable phylogeny, you can do keys that actually make sense. Classifications are not changing all the time. And for my work in USDA, a lot of our parasitoid work is in support of biological control initiatives in the classical sense, mostly biocontrol, and identification becomes a key issue for us. So that's kind of one of our main avenues to get this stuff right and not keep redoing stuff over and over again. From these keys and understanding kind of a better classification, then we can start to look at species. And I'm trying to make an argument here that I think it's probably falling somewhat on deaf ears here because we all kind of know this, but the better prepared you are taxonomically, the better you can address an issue when it arrives either in our forests or in our fields. And that's kind of part of what I call taxonomic preparedness. Right now I'm in the danger zone, like hardcore with cryptic species, and out of my comfort zone with cryptic species. And so I'm going to share what I've been doing with that and where I'm going to go next with it. And last, perhaps the most controversial topic all night, on type specimens and how I think, and a few of us think, we need to be doing more regarding type specimens. Okay. So, working in Hymenoptera since 1997, we've been pursuing a tree for the order that makes sense, that we can utilize for making keys or whatever. There was always not enough specimens. There was either not enough genes. We had to go get frozen tissues and all this sort of stuff. But we have this huge amount undescribed. By the way, this is the Vesta mandarinia here with the little antenna on it. This is what's going to be on display later on. We think the museum here has something like 3.5 million specimens. It's 50,000 species or so. We have a huge resource here. And one day, I'm walking to the restroom, and Sean Brady's walking the other way, and we meet midway in front of the collection, and he's like, "Hey Matt, we just got like sequence data, 1,000 loci from a 100-year-old bumblebee. What do you think of that?" I thought, that's awesome. And then as I was continuing my journey to the restroom, I started thinking, what would I do with that? And I remember turning and looking at the collection and thinking, if you could get that, then you could like sequence all these things in here, or at least some of them, and be able to do analyses without actually doing any field work. And they're already identified for you. That's a big thing. So we pursued, oh yeah, and there's a lot of small ones, and that's really where I live. So this little guy, this is Trichogramma KK. It's a huge biocontrol agent of a moth that eats wheat. You know, taxonomically super important and like impossible to see. So what could be better? So yeah, these are all the things that we kind of struggled against. Computational problems. These are all things that have kind of like become sort of old timey in a way in this current state of phylogenomic analyses. But the big one was this meeting with Sean, because what we did, we decided to write a little grant, and we went to the castle, the Smithsonian Castle, for some seed money to explore how we could pursue this more. And the other great thing was, in the Heim unit, we had this whole team of people that were just like, I'll do it. I'll find specimens and put them up there, and it was totally awesome. Like it was like we had like a party. We just kind of went through and found all the things that we needed. We made our FIMS, field information management system, to keep track of everything, and all the experts in all the fields added their specimens to the analysis, and we started doing the work at the LAB, which is like over there. And a lot of the groundwork was actually generated here. A lot of primers were invented here. A lot of the methodology was perfected here. It's now gone to a whole new level from where we were. But it was a really exciting time. And this is what we ended up with: 765 taxa from 87 families. We actually had like 1,400, but we had to limit it down because we wanted to do some different analyses looking at speciation rates along lineages, and we were pushing the computational limits of models, and they were going to take like 15 years to finish or something, so we had to pare it way down. So we got out groups from other groups. Yeah, I won't read all that all to you. Up to this point, previous phylogenomic analyses that were either anchored hybrid enrichment or ultra-conserved elements, this is all ultra-conserved element data, focused on kind of an exemplar taxon sample. They were either interested in Aculeata, the stingings, ants, bees, wasps, not including a lot of the parasitoids, or they had specific questions they were trying to ask. Like Peters et al. Was looking at like as many genomes as possible for one single taxon per family. And what we want to do is just kind of explode all that. Let's just get as many. We know all the problem groups because we're the experts in all these groups. Let's find all the problems, let's throw them all in there and mine the collection as much as we can. So we had a very rich data set, and there were some surprises. First off, parasitoids emerged as, I'm going to advance this real quick because I have a better tree here. Parasitoids, this is our summary tree. Parasitoids emerged much earlier we estimate, much earlier in the evolutionary tree of life than previously estimated. So we pushed it back to the Triassic. And then there was this really long period of time before any other life history mode within Hymenoptera kind of emerged. And when it did, we start to get social insects, stinging things that will hurt you, and then some odd return to herbivory, the pollen feeders, etc. But a lot of these stinging Hymenoptera, they're actually still parasitoids, or what we call predatoids. They have a very hyper-specific prey item that they select. So that trait, the behavioral trait of I'm going to find this one specific nutrient source and just stick with it. That's a really intrinsic part of the story of evolutionary development in Hymenoptera. And that length of time was a bit of a surprise to us. And then we wanted to try, the reason why wasp waste and stinger are on here, we wanted to test, because I would teach this in Heim course, the stinger, the wasp waste are key innovations for Hymenoptera. Well, we wanted to test that. And I'm not a mathematician, so I don't really know how these tests work. But as it turns out, these tests were kind of equivocal. Like we didn't see the smoking gun that we thought we would see. But what we did find, what we actually tested for, is there an accelerated speciation rate in any one particular group? I thought things like Acarcia super nasty aphylinid thing that has like 1,000 species all look the same. I thought that was gonna have a really accelerated speciation rate. Or a pantaloons, the Bracana that no one can tell apart. These are things that appear to be radiating very rapidly. No, none of those actually played out. The two groups that seemed to have, were kind of free, that actually showed any sign of having an accelerated speciation rate were the gall-inducing eurytomids, the gall-inducing cynopoids, and the herbivorous tenthroidenoid symphytons. So which are arguably secondarily phytophagic. So it looks like that return to phytophagy has something to do with these rates. And that got us a lot of flack from reviewers because they're like, well, you're the expert, you're a cynopoid expert. You put too many cynopoids in the analysis, but we hold for that. So that wasn't, no matter how many we took out, we still got the same accelerated rate. So that was curious. We didn't expect that. That was a really neat story that we're still going to continue on pursuing. What does that mean? We're not really sure. So to summarize this part of the talk, the key innovations just kind of weren't as impactful as we think, or they might have been impactful at some point and they're not really anymore. These are really difficult questions, by the way, computationally to ask. There's a lot of assumptions you have to make in these models, and I'm not particularly comfortable with that. So I'm still thinking these are key innovations. I just don't have any statistical or pseudo-statistical data to support that. It is interesting that the secondary herbivory thing happened, but number 4 is really my focus here. Number 4 exposed to me the 200 years of collecting that's been going on in Hymenoptera and how you can leverage that for new questions. That is like, I saw it happening and it blew my mind. So it was just really, really neat. And all the data, all the curation, thousands of person hours that have gone into that, it came to fruition with these phylogenies. And it's just, I think we're not unique in that way. There's other collections where it's just another reason why we collect and why we curate and why we spend time. Loris and I call it Curator Friday. We never get Curator Friday, but we try to like sort things out, make things nice, so we can go find things to do cool analyses in the future. Okay, so we're gonna move on to the next step. We've gotten a handle on all Hymenoptera. So let's move on to the cynopoids, which I work on, which are two kind of large groups. There's the big cynopoids, the macrocynopoids, and the small ones. And the small ones are what we commonly find out of galls or sweeping, even in the little pollinator garden. There's some trampy ones that like to eat little flies. They'll all be there. But these are the two kind of main groups that we've recognized within cynopoidology since 1992, 93. And these are what typical ones look like. They all kind of look the same. To me, these are beautiful, majestic creatures, but they all kind of look the same, I realize. But they have maybe different colors, but they're all kind of like 3 balls and wings. These are a little bit more spectacular. The Ebolias, there's some phyton parasitoids, so kind of showy, aposematically colored, and maybe the Hymenopterids are too. These are the more unusual ones that keep getting missed in phylogenies because they're too rare. The Ostrocynippidae, I argue, are the rarest Hymenoptera known, with 3 total specimens, period. They all happen to be in my office right now. Don't tell Australia. They don't want me to come. Eschaetoceros, they don't have any mouth parts as adults. Pycnostigmines are this weird African group. We don't know what they do, but they have a pseudo-pterostigma. And the Paralosines are a key group here, key for understanding all the cynopoids, and we'll get into those in a little bit. But these are the ones that, I'll just admit these. These are the ones that really kind of have made phylogenetics within cynopoids difficult to grasp. They're either too rare to sequence, so we're kind of doing morphology, and that's not really working. So these are the ones I wanted to focus on, if we could, with these newer UCE techniques. And then finally, we have a group of cynopoids that they are gall wasp-like, and they actually live in galls, but they are not actually gall wasps. They're attacking the gall wasps inside, we think. Galls are tricky. You can't see what's going on in there. You can only kind of estimate based on who's coming out and when, what's happening. So these were really key to having the phylogeny as well. And again, because we have the collection here, and I spent some time curating it, we actually have all these now. This is what sorting thousands of malaise traps will get you, the 10 harness you need for an analysis like this. So finally, it all came together. Well, now this will just get cold. So a couple of questions that have been in the back of my mind regarding cyanopoids is, where do gall wasps come from? They're nested deeply within the larger clade of parasitoid Hymenoptera we call the Proctotrypomorpha. That's the infraorder Proctotrypomorpha. Why gall? Why would you do that? You just had this awesome strategy exploiting everything under the sun that's an insect. Why would you go back to eating plant tissue? So I'm interested in how does that even involve? And the big question was, these phygotoid things, these things that are phygitids, that are entomologous, but they live in galls, are they going to tell us anything about this weird story? So these were kind of my focus. Most critically to this whole cyanopoid segment of this talk, though, is this bit right here. This is from our big UCE analysis. And right here we have the platygastroids, our sister group to the cyanopoids. This turns out to be the most critical thing I'm going to tell you tonight about cyanopoids, because all previous analyses that involved the kind of intricacies of cyanopoid phylogenetics never used one of these outgroups. They would always root on another cyanopoid that we thought was the most plesiomorphic, archaic looking one. That turns out to not be the best strategy. But we never really had a choice in the other. Previously, there was like outgroup of the day or outgroup of the hour sort of thing, depending on your analysis. There was no good outgroup to rely on. This one, the platygastroids, turned out to be really key. So this is what we know earlier about cyanopoid relationships as Ostrocynippidae, which we still haven't sequenced, by the way. That's on the to-do list. Is sister group to the rest. And then these macrocyanopoids are sister group to the remaining microcyanopoids. And that these macrocyanopoids were somehow more, deeper in the tree, plesiomorphic, if you will. I will make a note, though. I always make a note here. The first person to actually propose any phylogenetic relationships within cyanopoids was our friend Alfred Kinsey, who did these kind of area cladograms before there was such a thing. In his theory that gall wasps evolved out of northern Mexico and kind of spread through southeast United States. So that's pretty cool. I think it's pretty. So this was my first large attempt at a higher level analysis. I did this with Sean Brady, and this was actually publishing the last chapter of my dissertation that systematic biology refused to publish because the editor didn't believe in divergence estimates, period. So it got rejected. So I found somewhere else to publish it. I can't remember where I can't remember where Invertebrate Systematics, they took it. But I improved it since then. But you'll notice a couple things here. First of all, it's very fidgeted heavy. The only cyanipids are actually up here in the blue. And this green area are all things associated with plants, either galling, they're attacking gallers, they're attacking aphids near gallers, they're attacking syrphids, which are attacking aphids, which are feeding on galls. And then we have the eucoilins, which are all kind of more focused on being diptera parasites, dipterolarval parasites. I was pretty happy with this. At least it made some progress. But there were still a lot of holes in it. Mostly, there were very few cyanipids in there. So I was working with Fred Ronquist and Johan Lilleblad, former student of his, to build what we call the El Grande matrix. And technology moved forward before we could actually put that matrix together. And so that's where we are today. But right before that, the kind of penultimate kind of classic phylogeny, Sanger sequence phylogeny, was the 2015 Ronquist paper, which recovered a monophyletic gall wasp group, but a paraphyletic parasitoid group, which was pretty much counter to exactly what I found. So it appeared there was definitely a taxon sampling problem here. I think if you favored one particular group, it always came out monophyletic. So part of what I wanted to do was to address that. And we did that when, again, as a twig project of the all Hymenoptera UC analysis, we focused exclusively on the cyanopoids. And this was meant just as a kind of a dramatic graphic about our coverage. So the terminals along the right, those truly span the entire superfamily. Missing Austrocyanipede and the enigmatic Kwakwaini from South Africa. Since then, we've gotten Kwakwaini, but we still don't have Austrocyanipede. And this again was with Dietrich and Bonnie and Sean. And this is what we ended up finding here. Big highlights, the ebolids, the macrocyanopoids, and the other macrocyanopoids, the Lepterids, actually are quite derived. They're not the most plesiomorphic. They're not the sister group to all the cyanopoids, as we turned out. And that's a function of including an appropriate outgroup for the first time. I really want to, if I had the time, I don't know if it'd be worth it, but it'd be fun to rerun all the Sanger data with a Celionid outgroup and see if we get the same deposit. We probably will. But I have other things now, so I probably won't be doing that. The other big deal was cyanipids, the gallers, did not come out monophyletic. I didn't make, because this was like kind of prelim, I thought this was an awesome tree and I'm really proud of it, but I didn't think it was ready for prime time to start making nomenclatural changes. So I kind of just left it at that. But I will point out, we did estimate, that's what the tree here on the right, these little weird pie charty things are likelihoods of biology at those nodes. We theorized these paralosine, these weird galler parasite things that end up at the very, very bottom here, the sister groups of the remaining cyanopoids. We estimated them to either be parasitoids or inquilines, not phytophagous. So the earliest diverging group within cyanopoids we estimated to be either inquilinus or entomologous. We didn't have any data to what they're actually doing. They're on nothophagous galls in Chile. But we can get a lot of them if we fog, but no one's actually done the careful rearing of the galls themselves. Until these guys did it. Hearn et al. Just came out with this tree earlier this year. Jack Hearn was a student of Fred Ronquist's, and they did protein coding genes, and they actually got the same tree as we using a lot more data. They didn't include the ebolids or Lepterids, so that question is still up in the air with respect to our data. But they got confirmation. The paralosines are now paralacids. As you can see, they actually did name these groups, are parasitoids of Adetrochus chalcidoids, gall-inducing on nothophagous. So the earliest diverging group within cyanopoids is actually entomological. The next group being Diplolepidie, these are the rose colors. So it was nice. It's always nice to see some consistency there. Okay, so this brings us back to the question: well, how do these things evolve? And I spent, when I first got the cyanopoid trees, I was doing Heim course in Taiwan, and Donald Quick was there, and I took him aside one day, and I'm like, "Look, Donald, like, does it make any sense that you would be entomophagous and modifying your host's immune system, trying to hide, trying to manipulate it? Do you think you could co-opt those genes or some of those products of those genes and make it trick plants into growing galls? Like, does that make any sense?" And he looked at me, he's like, "Matt, how else would they do it? Like, that is the answer." And I'm like, "Well, okay, but we don't need proof." He's like, "No, we don't need proof." But and since then, we have found that venom glands of gall-inducing cyanopids are like twice the size of those found in their entomophagus sister group. So there's definitely an expansion of some of the anatomy, perhaps producing a lot more material that's injected when the egg gets injected, because we know when the egg gets injected, that's actually the beginning of gall induction. If you wash an egg and insert it, nothing happens. So there's something the mother's delivering with the egg. So we've gotten a little bit closer, I think, to what's happening here. And I presented this at this International Gall Symposium earlier in the year, and we all just kind of looked at each other like, yeah, that could be. Like, there's no real answer. But it's a really neat question, I think, to ask. And the other thing is just totally on the side here. In a presentation I gave at that symposium, the number of times gall induction has evolved independently in Hymenoptera is alarmingly high. 5 distinct evolutionary events across the whole order. Now, gall midges are the kingpins when it comes to gall inducing, something like 13,000 species, but it is a one lineage. Within a Hymenoptera, 5 distinct. So this is when my point is, this is kind of like rattling around in their genome. They somehow have the mechanism to do this kicking around. And I think that's actually will be a really neat paper, I think, to explore. And I think I might try that. We'll see. Critically, outgroup choice is huge on this. Okay, so let's move on to the next phase, and that is, what do you do with the stable phylogeny of cyanopods? Well, the first thing I thought of was, I've been using Hymenoptera of the world literally since I was an undergraduate. Like my training in Hymenoptera is because of this book. It's so easy to use, and it's the only holometabolous insect group for which there's a key to the whole damn order, no matter where you are on planet Earth. It's pretty amazing. Now, the classification has changed. Recently, chalcidoids doubled the number of families included within that group, and that's obviously not captured in Hymenoptera of the world, but there is a key couplet that'll take you to Chalcidoidea, and it will get you there every single time. My gripe was teaching the Hymen course, trying to use this. I kept telling Jeremy Frank, he knows the story. I have to tell everyone, okay, now this name, that's now a family. This one's now a subfamily. It got confusing. It got so confusing that people are like, what's going on? Like, you guys make up your mind over this stuff? And so I thought, starting in a Swedish offering of Hymen course, I had my Swedish colleagues that were like, let's redo the key. Let's make a new one. And we just grabbed images off Morphbank, and we just made a key. And that eventually got to this point in Taiwan where I had like the last version, and me and Chung Bi, who's unfortunately not in this image, I don't know, I couldn't find a single image now. He and I finished the cyanipid portion of this key, and we tried it out with the students. And that's published now. But before that, we had some attempts. The main one being the Afrotropical cyanopoid key. This was before the UC analysis, and the Afrotropics only have non-native gall inducer, because the quaquaine. That's the only one that's a native there. And there's this other thing called Roophilus that shows up as well. It's largely the entomophagus groups you find in Africa. And so me, Simon Van Noort, and my colleague Matthias Verschag, we met in Cape Town, spent a month, and just went through all the specimens. We have a colleague, Bob Copeland, in Kenya, and he's just collected so much material. I brought everything I had from here that was all mounted and labeled, and we sat there and just figured out a key. We like made a key to all these genera and tried to figure out how you tell them apart. Which ones are BS? There was a lot of BS ones. We're like, well, let's just kind of synonymize these or kind of move these over. So that helped get to the point where we could make the world key. I did a little one for the UAE. There's another one with Frederick on the tropics. And these are groups where you get to kind of the end of the couplets, and you're kind of left at an unhappy place. It's like a garbage bin. It's the last couplet. But at least the obvious ones we were able to pull out. But I tried to pull all that together with the new phylogeny and figure out how we could come up with a better world key, and simply just take a little knife and take the whole section out of Hymenoptera of the World and insert a chapter directly into it. And so this is what we came up with: World Cyanopoidea, key to higher groups. And it's already slightly out of date. We've now added 2 families that go to tribes, I think, at this point. And it's also available on the web at waspweb.org, which is Simon van Noord's website in South Africa, which, if you're into Hymenoptera, definitely check out that website. It is stunning. And the dude did it all himself. Like, I mean, he works with people, but he creates it. It's incredible. It's an incredible amount of work. And we wanted to make a key as user-friendly as possible. I thought, if a barrier to entry is, I can't tell these apart, let's take that barrier down. And then we get more people involved in study. It's more fun for everyone. I like photography, so it was a natural sort of thing. Boy, did it take a long time, long time, but it was totally worth it. But there was something that kind of came out of this as we're making the keys. First, I had no idea how hard a world key is to make. When you know in your mind all the collections you've been through and all the sorting you've done, all the weird exceptions to all the couplets. You're trying to make a couplet that is informative, yet manageable, because it's very easy to make unwieldy couplets where you're like, what? And so trying to work with students in hindquarters really helped. They're like, why is this couplet 9 characters long? Oh, that's what you got to learn to tell that group apart. Well, that doesn't work out so well. So in order to do all this, I figured out to generate a key at this level, you really have to figure out what your diagnosable. What are the diagnosable groups? Not just the groups. What are the ones you can actually diagnose? Which are the ones you actually have something you can wrap around? Are they stable? Do things have to key out multiple times? And ours we do. If it's a apterous gall inducer, which we have around here, it's a sexual generation, has no wings, crawls up the trunk in the middle of January. Those don't key out, and so you have to key out twice. Those are the diagnosable groups. And then you need to come up with character systems that people really, really can work with. The example I kind of drew from is Bolton 80, the key to AIDS. If you haven't seen that, that's a hell of a book. It looks like a coffee table book, except it's like scientific. It's totally awesome. So that's like how you build a key to a very, very difficult group. Ants are difficult. He made them easy. I thought ants were easy. I'm like, oh, look at this. Well, that's obviously didn't just spontaneously generate on its own. In this case, for these character systems, the stability, we started with genera. What genera makes sense? A lot of them didn't. And we just made note, like, we have no idea what this name means. No one knows what the type is. So we're going to list it. It's going to be part of the catalog, but we can't diagnose it. So we're not going to try to pretend like we know something that we don't. But this was the surprise to me. When you make a key like this is actually how you discover something. This is how you discover what are really diagnosable characters. Not what's in a diagnosis, like when you describe a genus or a species, but what are the things that actually make or break a couplet? You really have to test it. Then you find out how good your taxa are. And sometimes you have to go back and go, yeah, subfamily, it's kind of, it's not so good. No one can tell it apart. This doesn't always like win. It's not a win-win all the time, but I couldn't believe it. Matthias and I were sitting back like, man, we've actually like helped figure out the classification of cyanopoid moths. That was neat. I didn't expect that. And I think keys written for other people that made keys, which is like this weird, like old-timey kind of folklore sort of thing. I don't believe in that. Like that's kind of lame. We should make keys for people to really use and try to make them as user-friendly as possible. And go the extra mile, the extra photograph, whatever. Everything's digital now anyway. There's no more page charges in that way. You don't need like some big old thing. You can just do it you can just make it on the web. Okay. Next topic. I'm gonna just hide this again. So because we're these okay, we've gone from keys now to limiting taxa. So what do you do with these delimited taxa? What's the kind of end game here? And my end game is that when I work with people doing classical biocontrol or invasive species stuff, being able to give them, I'm only one person and they can send me specimens all day long, but sometimes takes me some time to get to them. So if I can give them tools that they can use, that's good. So using something like phylogeny gives some predictive power on what is going to be invasive or of potential use in a biocontrol case. But having some built-in taxonomic preparedness, some understanding of how many genera are in that subfamily. Are these genera actually backed by any characters? Those are good things to kind of know, and that's part of it. But I'm going to give you some cases here where that range from that was pretty easy to oh boy, we're in the briar patch. So the first one, this is the little brochure I passed around, is this thing, Dryochosmus curryphilus, the chestnut gall wasp. It's the only real pest cynopoid, if you will, because it attacks commercial chestnut. I got concerned because they want to, U.S. Forest Service is commingling commercial Chinese chestnut genes with American chestnut, which is functionally extinct from the landscape, to try to make them resistant to a blight that nails them, make them about 10 years old. Well, I thought if they can do that, then little curryphilus here might be able to find them because now it has the right genes, might be able to gall it, and then kind of undo the 50 or 60 years worth of work that they've been trying to put together to bring American chestnut back into forests. So in order to do that, I argued to the Forest Service, and they funded a project. We should figure out more about Dryochosmus curryphilus. Like, what is it in the right genus? Is it one species? How many invasions have there been? Like, what's the gene pool look like out there? If you're going to try to release a parasitoid against it, it's good to know these kinds of things. I said, sure. Okay, you got the person. So Chong Di was our postdoc here for a while, and he worked this up. And he came to me one day. Well, okay, this is the distribution. So Southeast Asia, native range. This thing is DK, as we call it, is a pest so severe in Italy, the government will come without asking you and destroy your chestnut trees right on your property. They will cut them down and chip them in your yard. So you get mulch, I guess. But that's kind of pisses some people off. But that's how desperate they are, because it's destroying their commercial chestnut growing adventures. So Chong Di came to me very concerned with this tree, that he sequenced the barcode, just the barcode, and ITS2, from all over the United States, wherever you can find DK, plus Japan and China, and he gets a rake. So he did it again. And then he did it a third time and kept getting, because I'm like, well, maybe, I don't know, maybe it's contamination. What we've determined now is it looks like there is just one CO1 genotype across all of these things, that this thing is just, and it appears to have jumped into the United States once, and it's just kind of kicking around out there. That was a little bit of a surprise. That's not what we expected. It'd be fun to do this with UCE data. We could probably get some structure out of it, but this is pretty amazing. The other thing he found was that Dryochosmus is a complete disaster of a genus, that it is a classic garbage can, and it's probably 4 genera. And that curryphilus definitely does not belong in the genus Dryochosmus. So this was an interesting kind of side project that the Forest Service funding provided, was the first insight into some generic level classification changes within Cynopene. And I will take a moment here. This is not the fault of the taxonomists working on cynipids. Cynipids are notoriously difficult, even at the genus level, because they all literally look the same. There's very little difference across a very large portion of an entire tribe. The taxonomy is based mostly on what plant is it gall, where on that plant is the gall, and what does the gall look like? If you have all that, you can tell your species apart. But if you have a wasp, Cynopene, like it's the tribe is actually what you can get to. And so this has led to a future project with Brendan, once he's established and set up. We're going to ask the question, why do all gall wasps look the same? Why is there like so little morphological divergence in this huge group that's actually, at least 100 million years old? What is that? That's a cool question. I mean, it's confusing for a taxonomist, but maybe there's something neat there. Okay, now we're going to move into a group I've been spending a lot of time on. And people on the hind floor heard me talk about this. I apologize if it's like, oh my God, is he going to talk about this again? This is the spiderling Drosophila scenario. This is Drosophila suzuki here, which is a drosophilid that thinks it's a tephritid. So it's got a little saw, and it lays eggs and fresh fruit, not rotting things like the rest of drosophilids, and it eats soft fruits. Billion-dollar industry. It eats them from the inside out. Can't spray them because they're inside. People making pies and stuff. They got maggots coming out of the pies. This little wasp here, this is Leptoplena japonica. It's a male, so it's not going to do anything at this moment. But this is one of the two players that are being released right now to try to knock this thing out in North America. It's another system that's Southeast Asian origin. Great photo. Just these are small. That's a 1.5 millimeter wasp right there. My pal up in Vancouver, boy, he knows his way around a macroplants. So we started working because there's a chalcid and a braconid. Me, Mike, and Bob have all collaborated on this project, and this is definitely a more confusing system than the Dracosmus. First of all, there's 4 species across 4 families. And they're all interacting with these hosts at different times. They're all attacking things at different rates. It appears that one uses another one to knock out the host immune system, and it takes it over. There's a lot of competition going on. And from a evolutionary knowledge, I find that fascinating. This is how speciation takes place. If you're a biocontrol person, like this sucks. Like you want it simple. You want to like knock stuff out quick. That's not happening here. But it all starts with a basic taxonomy. And a couple things, Leptoplena and Pachycropoides, these are fairly well-established genera. We know the limits of these genera. We know where the types are, that represent the genus, and most of the species in there. And then we got a mess. We got 2 things, Azobara and Ganaspis. Bring up Azobara to Bob, and he'll slowly close his door. He does not want to hear about Azobara. It's like, it's one of those. Ganaspis, I'm like, oh, I can solve Ganaspis. Well, that's turning out to be really difficult, and it's made even more difficult by a few other things I'll share here. This is the general life cycle. It's another tiny litwack piece of art for our outreach purposes. So the parasitoid is hunting for its host in the fruit. It's having to pierce the fruit, pierce the matrix inside the fruit, poke around, find the host, lay its egg. The egg can either hatch into another wasp, or if it's the wrong life stage, it can be encapsulated. It's the wrong species, if the wrong parasitoid hit it, it might get stabbed, but it can encapsulate the egg. Or it may go unparasitized. It can hide, they can escape. If the fruit's rotten a little bit, it can kind of swim away. So these are the 2 different scenarios that we're trying to work out here. The Ronin wasp, as we call it, because we're all into common names now. So the Ronin wasp is part of this genus Leptoplena that my academic grandfather sorted out in the 70s. And he literally, there was no other cynopoid worker back then, none alive. And he just like, I'm going to figure this out. And he kind of sorted out the genus Leptoplena. And because of that, as additional species were discovered through rearing, when early barcode data first started coming out, this thing Japonica was described maybe 15 years ago. So it's a fairly recent one. And it fits, we can key it. It phylogenetically makes some sense. And it's a big time parasitoid of Spiderling Drosophila. This is a key, a phylogeny I did, more exploratory. This isn't published. This is all Japonica here. What I do is these folks go to Korea. These ARS folks go to Korea and China, and they rear out, I kid you not, 30,000 puparia at a time, and they save and link everything that reared out of a puparium back to its original host plant. They record it all inspection. So when these things get sent to me, it's overwhelming how much time they have spent. So I go absolutely the extra mile, and I've sequenced so many of these things trying to figure this out. That's why there's so many Japonica. That's why you get this rate. So I was curious, like, what's the topology look like within Japonica? Are there cryptic species or something weird going on there? So far, we're keeping it just as Japonica, but this is kind of our best case scenario where we actually had some idea of where this taxon might go, and now we can actually put a name on it, be able to figure out diagnostic characters. Okay. Ganaspis, not even close, pal. It's 67, we think 67. We actually don't even know. We can't figure out how many species there are in the genus. We have no idea where most of the types are. Classic garbage can. I've seen gall wasps described in Ganaspis. It's that bad. It's totally polyphyletic. So I thought, oh, I can kind of figure this out. I can at least figure out the obvious ones. And sure enough, the thing that they were rearing out that was really common, that was not Leptoplena, was what I consider as a practicing taxonomist of cynopoids, I called it a Ganaspis. It fit my concept of Ganaspis and the concept of Ganaspis that I had put into keys for genera. What? Okay. This plagues me. I have this hidden audio tracks I cannot get out of some of the slides. I bet it's only this slide. The problem I was having was the folks that were doing the rearing. They were finding different CO1 haplotypes, and that was linked to different host rearing conditions. So if they reared spotted-winged Drosophila from hosts on fruit found on the ground, that was a different haplotype than from fruits infested with spotted wing still on the plant. And the ones on the plant were the ones that we're mostly concerned with, because that's the fruit that hasn't been harvested yet. You don't harvest fruit off the ground, you let it go, you ignore it. But stuff that is still on the plant, that's what you want to hammer with your natural enemy. So they kept telling me, like, well, and they don't interbreed. They did some rearing work in the lab. They wouldn't interbreed, and they ended up with this nomenclature of G1, G2, G3, G4, G5. These 5 Ganaspis lineages that I called all this thing called Braziliensis, which isn't even from Brazil. That's from Panama. But I don't know. Someone decided to call it Braziliensis. We had the type in the collection, so that's the name we've been going by. They kept telling me, I don't know. There's something else. There's something else going on. So I went back to Stockholm, which is kind of my intellectual roots with these, and met with Matthias. This is Matthias here. We took a moment to go record on. And Frederick, he gave me some ideas, and went back and started looking at all the morphology all over again. I was able to pull out one lineage, this thing that was called G5. I was able to find stable morphological characters on this scutellum that I could separate it from G1 and G3. And that, as it turns out, matched perfectly with the type specimen Braziliensis. So that left us this G1, G3. What the devil do we do with these? Oh, okay. So this was our original barcoding tree.
Share some files.
Zoom share. High control. Okay. So again, like the Leptoplena situation, the Ganaspis came and they were reared. I literally had like 9 full drawers of these specimens, each with the host remains into it. That's how it's the gold standard for what we work on. I barcoded as many of these as possible just to get an idea, what does it look like? And sure enough, I got this G1 thing and the G3 thing. And then G5 was a conglomeration of stuff in colony that people were calling all kinds of names. And I was able to put them all together into one group and start vouchering these things. Just as an aside, these types of fidgeted wasps are super easy to rear in the lab, and they're used as kind of lab rats because you can rear a couple hundred in like a little tray under ambient conditions. But they're small, and I can't tell them apart when they're alive. And so names kind of floated of what were in vials. They also cross-contaminate each other. So I wanted to voucher all these, link them to actual specimens, and figure them out. But I'm not entirely convinced within cynopoids that CO1 is the most reliable for looking at evolutionary relationships. So I wanted to take it up another notch. I wanted to take a look. So again, we're going to do UCEs again, but this time kind of at the species level. Leading up to this was a genome analysis that I did with the folks up in Newark, Delaware, and looking at the different populations from different parts of China and Japan and British Columbia, because lo and behold, there's so many infested shipments coming from Asia that both the Leptoplena species that we looked at earlier and this Ganaspis, they've arrived on their own. They're here doing the work without going through quarantine. They're just kind of hanging out, doing their work. But that's kind of not great. We need to do some post-range testing before we release things. And when they come over on their own, that's kind of that's a problem. So so Don and I published a paper with some other authors, this book chapter on unintentional biocontrol, and that's exactly what happened here. But my colleague up in Newark, he looked at this genomic data. He's like, these might be like different genera. Your G1, G3, that you can't tell apart. They're that different at the genome level. I was like, okay, now it's mounting. It's mounting. We've got two things that are biologically behaving differently. They don't interbreed with each other under lab conditions. Their genomes are so different. The expert says they might be different genera. Looks like we've got cryptic species here, and we got to take care of this. So I got Jeff Sosa involved in this. He came to me one day. He's like, "Hey Matt, do you have any projects that are like super practical that we can like knock out?" I'm like, "Hey, I'm looking for someone to do like genome stuff of cryptic species. You know anyone who does that?" So we like over a coffee agreed to do this project, and this is what he came up with. So this is doing UCE data we took all the extracts that we had already made for the barcodes and just reran them again, this time for UCE data. And then he ran these statistical tests, how many species do we have? Fred Ronquist suggested we do this as well. Well, he asked these computational things, 60 to 85 cryptic species. I find it hard. So I said, no, we're not gonna believe that. We're gonna just stick with our G1, G3, G5. That's good enough for me. There might be, given the topology in here, one could argue there's more cryptic species in the G5 group, but they're not part of the biocontrol scenario, so they're less critical right now to get names. Because what we want to do is, if we're going to name these things, they've got to be named yesterday, so the biocontrol community can provide a valid name and not be referring to population. It's APHIS that oversees whether or not you release something. They want a name. They want to know an expert's like been involved. It's not just like ASBS. So you know, I get it. That makes sense. So I've been putting on my effort. But this is just to show kind of what it looks like. And this is this is the tree I'm preparing to submit like right now. It actually is public. I just had to fit in the screen. So G5 is largely this Braziliensis, the original name at 1905, links to a whole site. G3, I'm calling Lupanii. And then G1, the real action packer sort of major natural enemy, that is Chimorum. And Chimorum is, I'll slide what that name means. And then what I've done is I color coded all the different collecting events, all the different folks that provided specimens.
Yeah.
Can someone turn their mic off out there in TV land? Shovel. Okay.
I'm gonna go to the bathroom.
Pretty quiet there.
This meeting's over.
So what I wanted to do in here is not only match or map all the different collecting events and the colonies that were derived, so people know, oh, my colony is here in the tree. But I also, these are the holotypes. There's this one here for Lupanii, and then the Chimorum is right here. So it's important to me as a morphologist working on a cryptic species, like, how do you do that? Like, they all look the same. So I had to at least make sure the holotype landed where it was supposed to, and I could kind of use that as an anchor. We'll talk more about anchors in a little bit. So this is kind of my wrap up for what I found here. G5 is this pan-tropical thing. It's kind of a tramp. It doesn't even attack spotted Drosophila. It's just a Drosophilid parasitoid. Chimorum, which is a combination of Kim Homer and Professor Kim Murrah from Hokkaido. Kim Homer is a colleague of mine, just retired from ARS. So it's a mashing up of their names. This is the one that's going to be the big biocontrol agent. And Lupanii, for those in the mango world, this is based on Lupin the Third, because it's G3 and it's Lupin the Third. And Lupin the Third, while he's a criminal, he tries to do good, and that's what Lupanii is trying to. It knows what it's supposed to do, it just doesn't want to do it. So it sometimes parasitizes the right thing, sometimes it parasitizes wrong. So Lupin the Third is super entertaining if you want to get it. And it does appear that CO1 is fine for diagnostics. We actually have now vouchered specimens linked to authoritative, clean sequences in our Drosophila parasitoid database drop. And so people can now not have to send stuff to me. They can actually query the database and find out what they have. But unless they find something that doesn't fit the database, then they send it to me. Because there's going to be surprises out there. But at least we have a baseline now. So that's how long it takes. That's been since 2015. And I'm just now getting the paper ready to submit like tomorrow. That's what taxonomic unpreparedness gets you. And that's me kind of working on this a lot, like trying to figure this out. Those are the kind of the two extremes. So here we go. We'll just skip that. So the ovipositor clip. This is another kind of piece of artwork trying to interpret this particular structure, because I do think we found a single morphological feature to separate G1 and G3. And it has to do with this notch subapically on the ovipositor that allows the ovipositing female to lock her ovipositor into the cuticle of the host just long enough to keep the host from escaping, because they will escape. It locks into the host, and then she can inject a small amount of venom that calms the larva down. If she can't get her ovipositor out in time, there's like a learning technique or learning phase where they have to go through. If the female attacks the host that's too big, the host will start rolling away. They do this tumble technique, and it actually will pull the whole ovipositor right out of the female and drag the female into the growth medium, and they die. So there's a cost to having this thing. If you know how to use it, it's very effective. If you don't, it'll kill you. As it turns out, Chimorum, which is attacking a host in fresh fruit, has what I call a captive audience host. Since it's in the fresh fruit, it's almost a gall. It's the host is in there, and it can't really go anywhere. There's a little cavity. It's kind of carved out. It's eating around. It's living the high life. But it can't really escape like you can if you're in a rotting fruit. There you can literally swim through the matrix of rotting fruit and get away from this. So you'll need if you're in parasitoids, you need to restrain that, keep that from happening. But if you're in the fresh fruit, not so much. There's nowhere for it really to go. And on top of that, you have to puncture fresh fruit skin and then get through the matrix inside to find your host. I'm interested if that clip actually might even interfere with that. Like it would actually get caught, if you will, on the fruit of the skin, on the skin of the fruit as it's going in. And I compare this to a group of phygitids that are exclusively leaf miner parasites, agromycin parasites. And these are ovipositing through leaf tissue directly to their host. And not a single one of them have an ovipositor clip. All the rest of the phygitids that attack free living Diptera all have a clip. They all need to restrain their host, it seems. So this is my working hypothesis, that the small clip, if you get a wasp, and you can dissect out the ovipositor and get the light exactly right to the point where there ought to be a clip right here, but I can't see it, that's actually Camorra. It actually is a clip, and it's so small. It took me 30 or 40. I thought I was mounting them wrong. I was like screwing around, you know. Turns out its clip is just really small. And the Luponia has kind of a normal one. So this is the feature I found that can tell these things apart. But it's not even quantitative. It's more like a feeling. So this is where CO1 will be very useful for discriminating these groups. Okay, so to summarize this section, I think Crypsis is cryptic species. It's a relative thing. Like, ask someone 100 years ago, some of the phygitids I see all the time now, they're not cryptic. I can easily tell them apart. But if you've got like bad optics, bad light compared to what we have now, they are going to look cryptic. So it is a relative thing based on the technology. Just because I morphologically can't tell them apart, we can genetically tell them apart. How cryptic is it really? So something that I kind of taught myself about this is a way to kind of look at that. And this is where I love this particular position I have, because to have people rear out tens of thousands of hosts, that just doesn't happen in the groups I work with. So this was really, that's why I put so much effort into this. It's been really, I like to put as much effort as they put into it, and in my work with them. Okay. Now, this is just total discussion fodder, but I'm going to make some points. This came out of some long discussions. Some might have involved people being inebriated. It was definitely late at night. It was definitely the end of long days at ECN and ESA at our lovely National Harbor. And that is on types. And what do we do about types? There's a lot of discussion, and I think it's perfectly fine discussion about how come types for certain places are here and not where they were collected. I'm not going to discuss that. There's a lot of discussion about that. I'm going to focus on why we haven't digitized all type specimens yet. Why haven't? Why hasn't it happened? And what are we missing by that not being done? And the term taxonomic drift came to, I don't know if someone else came up with it. Loris and I were discussing it on the way into work one day. Because we're actually making notes. So a lot of this came from notes from that car ride, Clara Barton Parkway. I deal with taxonomic drift in GenBank, where a technician in a lab has put a name on a vial, that the members of that vial get sequenced, and that name gets associated with them in GenBank. And that's it. That's what it is. These are names that I've looked up names where I've never seen that name before. I can't find where it came from. Or I've seen names where no one has ever found the type of that name, the type specimen. So there's no way to know how did they possibly come up with that name? Like, I don't even know what the type is, and I like work on the group. Like, so there's that kind of drift, and we created a Drosophila parasite database to manage that. But then I started thinking more about this concept, that we have things in the collection that are being digitized right now at the species level that are identified. They have names on them. I wonder how far off they could be from their original species concept that the author of that taxon meant. You get specimens that say CWT, compared with type. I've seen compared with type like totally wrong. Like I've seen that type. Doesn't look like that. Not even close. Like that's just my little group that no one really pays any attention to. I'm wondering if how common this is. So what do we digitize if those names could be wrong? The only taxa I would argue in this collection, the insect collection, the only taxa that are absolutely what they say they are the type specimens. Even if they're like a synonym, they're at least what that person said. We have tools now that make this easier than it's ever been. It's really a labor issue. And that's where I drug. Okay, let's go into this. So I curated home, curating the record collection, and I come upon this album cover, which is always fascinating me. And it just dawned on me. This is kind of like what I was trying to think of a graphic. What kind of graphic do you associate with this taxonomic drift and types and CWT and all this sort of stuff? Well, from a distance, this graphic on the cover, it looks like a homogeneous kind of like a unit. There's space on either side, and that could represent a character distribution of what we would call a species. But then you look closer, zoom in on this area right here, and you start to see like a lot more structure. You actually see, this is the joy in this division, you see a valley forming here. Things they're moving apart. And I started thinking, if your type specimen is on one of these, and you could sequence that, if you make it knowable, then you have a reference point for everything else that might be around it. But if you don't have that, you could be at a very close neighboring peak. We used to call these little islands of trees back in when I was in grad school, little optima, little local optima. Any of these could be where you could be stuck on one of these. But if you have the type, which might be 100 years old, which we can now at least partially sequence, then you can reference everything else around it and make it knowable. Vetting these is a big part of it. That's a lot of labor. Is what we have in the type collection actually a type? Have we found all the types? I'm still finding types. I found a type specimen in the Hymenoptera teaching. This is a cynopoid, who cares? Well, I found it, I put it back. We have unit trays for which we've just lost the types. We'll never see them again, probably. So that's obviously not a vetting problem. Digitizing, in my mind, is making all that data available through like something like EMU, but probably more critically, taking a picture of the labels, and then we don't have to ship things out. People can see things a little bit better. We're documenting. This is all stuff we've totally talked about. I'm not pretending that this is anything new. This is, I'm glad Paul Goldstein's here. This is an idea I stole from him. I'm curious about the centroid idea, that if you have a semaphoron, you call it, based on a type specimen, as you kind of sequence in a rotation around that, then you can start to find, that's how you're going to find cryptic species and root them to an actual name. But until we get all of these digitized, that's not possible. The resulting species from something like that exercise are going to be more stable, and they're going to generate more stable genera, which are going to generate more stable higher classifications, better keys. All the stuff I've been talking about, all the way to the ordinal level. So this is my challenge. Before anything else is digitized in the National Insect Collection, I argue we should have all the types digitized, all of them, every single one. Things we think are types, things we might think are types, And I went, after coming up with this kind of thing here with Lourdes, I'm still dragging her into this, by the way. I went to Floyd and said, hey, could we estimate how much it costs to digitize the remaining Hymenoptera holotypes? Because we got some money for the bees at one point, got some money for the ants at another point. We got money for the authors that begin with the letter A, you know, so we had these like, and finally we had all the money to do all the rest. That was a $250,000 grant from Smithsonian to do this. It's a collection improvement grant or digitization grant. And while Floyd was very wanting to help, this is a very difficult question to answer. I wanted to get an idea per type specimen, 5 images. Those images being edited, those images with a scale bar, and then the time it takes to upload it. Could we get an estimate? And is this even remotely equal to a calculation error in the US military budget? And my guess is it's well below that. This is expensive from a labor standpoint and kind of nothing else. I would really like to see this. I think we all really would. And we're gonna put together a paper that kind of points this out, taking it away from the political side of it, whose property is biodiversity and where do these types belong, but making it more like, this is the scientific infrastructure that we desperately need. It's not high tech. It's not running robots on Mars. This is like just taking what we have, taking good photographs of it, and putting it back. I know it's expensive. I know it's like labor involved, but I think it's just really. And then Brendan brings up cyber typing. That's the next level. That's the next level. We're not there yet on an industrial scale, but I would love to see. So we're going to put this paper together. We'll see if it gets any traction. See if it. So this is my overall conclusions. I absolutely adore working here. This is like a playground every day, and my family like actually supports this, which is awesome. This organization is critical to my being.